Okay, let's dive right in. Today, we're tackling a drug that's, well, absolutely vital in the fight against cancer Taxol.
Hmm.
If you or, someone you know has faced breast, ovarian, cervical, or lung cancer, you've likely encountered this powerful chemotherapy drug.
Oh, for sure.
It's a real cornerstone of modern treatment.
It really is. But for all its importance, Taxol has this significant Achilles heel: how we produce it.
Right.
The current methods are, frankly, incredibly difficult, astonishingly expensive. We're talking upwards of $20,000 per kilogram.
Wow. $20,000.
Yeah. And honestly, not great for the environment.
Mm-mm.
It relies on this really complex chemical process
Uh-huh.
that starts with compounds painstakingly extracted from the Pacific yew tree.
And extracting from a slow-growing tree is inherently limiting, right? That sounds tough.
Exactly. It's a bottleneck. And this is where our source material comes in.
Ah,
We're
okay.
after a fascinating recent breakthrough. It's from a Danish research team at the University of Copenhagen. And it's detailed in a paper called Breakthrough: Taxol Biosynthesis in Yeast.
Biosynthesis in yeast. Interesting.
That's right. This team, it looks like they've finally unlocked a biological secret, something scientists have been chasing for decades.
Which is...
How the Taxol molecule is actually built step by step, in nature.
Okay, so our mission in this deep dive is to unpack exactly how this team cracked that mystery,
Mm-hmm.
what their new method using yeast means in, like, practical terms,
Right.
and the potential ripple effects this could have for accessing vital cancer treatments and producing drugs more sustainably, globally.
Yeah, globally is key.
We're going to help you understand the core of this discovery and why it could be, well, a game changer.
Definitely. And, for over 30 years, this has been a global scientific quest.
30 years.
Yeah, scientists worldwide searching for the complete recipe book, the natural biosynthetic pathway
The pathway.
that organisms use to create Taxol.
Mm.
Because the molecule is so complex, understanding this natural process, it's been seen as a kind of holy grail in the field.
A holy grail because knowing the natural steps could potentially lead to a much better, maybe more efficient way to make it,
Exactly, compared to trying to piece it together chemically.
which sounds hard.
Oh, it is.
Yeah.
Chemical synthesis is hard, complex, and very expensive for a molecule like this. Nature builds it elegantly, but unlocking how nature does it
Yeah.
requires identifying every single enzyme involved in that multi-step assembly line.
Okay.
And the Copenhagen team found the final two missing pieces.
The final two. So parts of the pathway were known already, but these last couple of steps were the key.
Precisely. And these weren't just any steps. These specific enzymes, they're crucial for adding the, let's say, finishing touches that make the Taxol molecule biologically active,
Yeah.
ready to fight those cancer cells.
So without these final steps, you might have something like Taxol, but it wouldn't actually work as a drug.
Exactly right. You'd have a precursor molecule, maybe, but yeah, it wouldn't work.
Okay, so they mapped the entire natural pathway. That's huge, scientifically, just on its own.
It is a major achievement.
But they didn't stop there, and this is where it gets really interesting, you said.
This is where it gets very practical. They took the genetic instructions, the genes from the yew tree,
Mm-hmm.
the genes that tell the tree how to make all of the enzymes needed for Taxol production,
Okay.
cloned them, and then did something quite remarkable.
Which was?
They inserted those complete sets of instructions into, well, humble yeast cells.
Wait, yeast? Like baker's yeast? The stuff you make bread with?
Huh? Well, related, but specifically engineered yeast, of course.
Right, engineered.
By giving the yeast these genes, they essentially gave the yeast cells the full blueprint and the necessary tiny biological tools to build the entire Taxol molecule from simpler starting material,
Just like the yew tree does.
just like the yew tree. The source aptly describes these engineered yeast cells as microfactories.
Microfactories? I like that. It paints a really clear picture.
Mm-hmm.
So instead of relying on labs full of complex chemicals and extractions from trees, you're potentially just, growing the drug in fermentation tanks using yeast?
That's the vision, exactly.
Okay.
And the implications are significant, starting with cost.
Right, the cost. You mentioned it was high.
Very high. The source makes it clear that this yeast-based method already allows for cheaper Taxol production compared to the conventional semi-synthesis approach.
Cheaper from the get-go, even before they've really refined it. That's impressive.
It is. And the team thinks they can do even better.
How much better?
Much better.
Yeah.
They anticipate that once they fully refine this yeast process and scale it up,
Yeah.
they expect to cut the production cost by half.
Half. on a drug that currently costs over $20,000 a kilogram.
That's the projection.
That's a staggering reduction. And the source explains exactly why lowering this price point is so incredibly important right now.
It really does.
Like, look at ovarian cancer, for instance. Projections show this massive global increase by 2050, prevalence going up over 55%, and tragically, mortality expected to rise by nearly 70%.
And what's really critical to understand there is that the vast majority of those projected increases, they're expected in low and middle income countries.
Ah, okay. So the places least able to afford the current price.
Precisely. The current high price of Taxol is a huge barrier there. This discovery, it offers a real pathway to making this essential life-saving treatment accessible to millions more people who simply cannot afford it today.
So it's not just about the price tag in isolation. It's a direct link to global health equity.
Absolutely. A huge link.
But it's also about sustainability. You mentioned that earlier. How does this yeast method stack up?
Oh, much better. The current chemical semi-synthesis method is resource intensive. It often involves using harsh chemicals and solvents.
Right. Not great stuff.
Not great stuff. And they have environmental downsides.
Yeah.
This new yeast-based method largely avoids these problematic inputs.
And I think you mentioned it can use different starting materials, too.
Yes, that's another key point. Instead of needing these highly purified, very expensive precursors for the chemical process, the yeast can potentially utilize less purified, cheaper extracts.
Yeah.
Maybe even taken from renewable sources like yew needles, not just the bark.
Oh, so that reduces pressure on the trees themselves even more.
Exactly. Plus, another benefit mentioned is that the materials used in this new bioprocess, many of them can potentially be recycled.
Okay, so multiple wins on the sustainability front.
Seems like it, yeah.
So if we sort of pull back and look at the bigger picture here, this research demonstrates something the source indicates is quite rare today. A biotech method for producing a complex drug that is both sustainable and low cost.
That combination is the key.
Why has that been so difficult to achieve historically? Is it usually one or the other?
Often, yeah. Biotech production, while it can be cleaner in some ways, can still be very expensive to scale up. Complex infrastructure, specific growth media for the yeast or bacteria, purification steps, it all adds up and drives costs high.
Or the cheaper methods aren't very green.
Or sometimes, methods might be cheap, but still rely on unsustainable inputs or create different environmental issues. This team seems to have found a really promising path around some of those common hurdles, at least for this particular molecule.
That really highlights the significance, then. It's not just a new way to make Taxol. It's almost a proof of concept, maybe, for a potentially new paradigm in drug manufacturing.
It could be seen that way, yes.
Yeah.
A strong proof of concept.
So what are the next steps? Where do they go from here?
Well, according to the source, they've already applied for a patent on this method.
Which makes sense, protecting this valuable intellectual property.
Absolutely. And they're actively working on launching a spin-out company.
A spin-out to commercialize this.
Exactly. Specifically focused on bringing this biosynthetic Taxol production method to market.
So it's moving from the lab bench towards, hopefully, real-world application. That's exciting.
Right. It is. It's a crucial step.
And the paper detailing this breakthrough, identifying the full pathway and the yeast method that was published in Nature Synthesis on April 30th, 2025.
That's the one.
Yeah.
A very recent and, I think, very impactful publication.
Okay. So what does this all boil down to for you listening right now?
Well,
This discovery offers a clear, tangible path towards a supply of Taxol that could be significantly cheaper, far more sustainable, and critically, much more accessible for the millions of people globally who need this life-saving treatment.
They didn't just solve a decades-old scientific puzzle by mapping the full natural biosynthetic pathway, which is amazing in itself.
Right.
They took that fundamental biological knowledge and successfully translated it into a practical, potentially scalable biotechnological production method, using yeast as the workhorse.
It's pretty incredible stuff, and it makes you think, doesn't it?
How so?
Well, think about what this kind of breakthrough in biosynthetic production could mean, not just for Taxol, right? But for other complex, expensive, or maybe environmentally challenging natural compounds.
Yeah, good point. Things used in medicine, agriculture, maybe even other industries entirely.
Exactly. How might this reshape how we source and produce vital materials and treatments in the future? Something to chew on.